Welcome to Biotechnology World. Now we are going to discuss the topic ligand designing. Definition: ligand designing, also known as rational drug design, is the process of designing molecules, that is ligands, that specifically bound to a target protein or receptor to modulate its activity. Key concepts: target identification. Identify a specific protein or receptor involved in a disease process as the target for ligand design. Structure activity relationship SAR. Understanding how structural modifications of a ligand affect its binding affinity and biological activity. Docking studies. Computational simulations that predict the binding mode and affinity of ligands to the target protein. Pharmacophore modeling, identifying the essential features such as hydrogen bond donors, acceptors, hydrophobic regions required for ligand binding. Ligand designing methods. De novo design, building ligands from scratch using computational methods, often guided by knowledge of the target structure and pharmacophore. Structure-based design, utilizing the three-dimensional structure of the target protein to design ligands that complement its binding pocket. Ligand-based design, designing ligands based on non-ligands or pharmacophore models without necessarily requiring the target structure. Ligand designing major tools and software. Schrödinger suite. It's a comprehensive suite of software for structure-based drug design, including tools for molecular docking and de novo design. MOE, Molecular Operating Environment. It's a software for molecular modeling, including ligand design, pharmacophore modeling, and virtual screening. Autodock, tool for molecular docking studies used to predict the building modes and affinities of ligands to target proteins. PyMol, visualization software often used to analyze protein-ligand interactions and design ligands. Major approaches. Fragment-based design, designing ligands by assembling smaller molecular fragments that bind to different regions of the target protein. Virtual screening, high throughput computational screening of large compound libraries to identify potential ligands for further optimization. Combinatorial chemistry, generating libraries of diverse chemical compounds to explore a wide range of chemical space and identify lead compounds. Major applications of ligand designing. Drug discovery, designing novel therapeutics to target specific disease pathways or proteins implicated in disease. Lead optimization, improving the potency, selectivity, and pharmacokinetic properties of lead compounds through iterative ligand design. Fragment-based drug discovery (FBDD), identifying fragment-sized molecules that can be elaborated into high-affinity ligands through optimization. Ligand designing validation. Experimental validation, testing the binding affinity and biological activity of designed ligands using biochemical assays and cell-based assays. Molecular dynamic simulation, performing simulations to validate the stability of ligand-protein interactions and assess ligand binding kinetics. Ligand designing key terms. Ligand, it's a small molecule that binds to a protein or receptor to modulate its activity. Docking, computational simulation that predicts the binding mode and affinity of a ligand to target protein. Pharmacophore. Say hypothetical three-dimensional arrangements of features essential for ligand binding and activity. Lead compound. Initial compound identified through ligand design that serve as the starting point for further optimization. Major challenges. Selectivity. Designing ligands that selectively bind to the target protein without off-target effects. Bioavailability. Ensuring that designed ligands are orally bioavailable and have suitably pharmacokinetic properties. Synthetic accessibility. Designing ligands that can be synthesized using practical and scalable synthetic routes. Resistance. Addressing the potential for resistant development by pathogens or cancer cells against designed ligands. Thank you.